Scanning electron microscopes. So, by definition of electron microscopes in general, it is used to analyze cell components and observe organelles. Electron microscopes, in specific, use electrons to form an image. And so this greatly increases the resolution of electron microscopes compared to optical, also called light microscopes, giving a more detailed image. A beam of electrons has much smaller length than the wavelength of the light, so an electron microscope can resolve or distinguish between two objects that are extremely close together. That's why an electron microscope gives a greater resolution than the light microscope. Taking a look at this picture, you can see that this picture taken by a light microscope shows the structures, but not significantly and not clearly, while taken by an electron microscope, you can see the structures very clear. So this is the nucleus, and this is the nucleolus. These are surrounding organelles in the cell. Magnification and resolution are two scientific terms that are very important to understand and distinguish between. A magnification tells us how many times bigger the image produced by the microscope is than the real-life object that we are viewing. So magnification tells us how many times bigger the structure that we're studying than the structure in real life. While resolution is the ability to distinguish between objects that are close together. Example, the ability to see two structures that are very close together as separate structures. This means that the electron microscope has a maximum resolution of around 0.0002 micrometers or 0.2 nanometers, which is around 1,000 times greater than that of the optical or light microscopes. This means that the electron microscopes can be used to observe small organelles in the cell, such as ribosomes, endoplasmic reticulum, or lysosomes, which are structures and organelles that cannot be observed using a light microscope. Now, there are two types of electron microscopes, the transmission electron microscopes and the scanning electron microscopes. A scanning electron microscope scans a beam of electrons across the specimen. So a beam of electrons hits the specimen, and this beam bounces off or back to the microscope off of the surface of the specimen, and the electrons are detected and so forming an image, meaning that they produce the three-dimensional images that show the surface of the specimens. However, these specimens should not be alive. They should be dead, and so they cannot observe live specimens, unlike the optical and light microscopes that can be used to observe live specimens. And they also do not produce a colored image, unlike the light microscopes.